Museums Victoria respectfully acknowledges the traditional owners of the lands where we work, the Wurundjeri and Boonwurrung peoples of the Eastern Kulin Nation, and First Peoples language groups and communities across Victoria and Australia. We pay our respects to their elders past, present, and their emerging leaders. Our organisation, in partnership with the First Peoples of Victoria, is working to place First Peoples living cultures and histories at the core of our practice.
Well, welcome everyone. My name is Dermot Henry and I'm the head of the sciences at Museums Victoria's Research Institute. And it's my great pleasure to welcome you to Melbourne Museum tonight for this special event, and a warm welcome also to those joining us online tonight. I begin by acknowledging the Wurundjeri, Woiwurrung and Boonwurrung peoples of the Eastern Kulin Nations, the traditional owners of the land on which we work and where we are gathered this evening. And I pay my respects to their ancestors and to all the elders and First Nations people who are with us this evening. I'd also like to thank Linda for providing Auslan interpretation for tonight's program. So thank you for being here, Linda. And so tonight's event is part of the Future Forum's Ideas Shaping Tomorrow program, and a series of monthly conversations proudly presented by Museums Victoria that brings together the world's brightest innovators and creative thinkers to reflect on the big ideas and important questions that shape our future. Today's program, Reversing Extinction, builds on the momentum of recent media attention and discourse surrounding the de-extinction debate. It comes at a timely moment as this week, the federal government launches their Threatened Species Action Plan. It's called Towards Zero Extinctions, following the devastating assessments in this year's State of the Environment report. We are delighted to be hosting this event. For over 165 years, Museum Victoria has been collecting, identifying and recording the biodiversity of our region. Our collections contribute to a baseline for measuring environmental change and biodiversity loss since European settlement. They support conservation research, and they drive groundbreaking projects to protect critically endangered species and ecosystems, and we'll hear a bit about that tonight. At the recently established Museum Victoria Research Institute, we are undertaking world-class wildlife genomics research to inform conservation strategies for threatened species. We're working in collaboration with government instrumentalities, universities and community partners across Australia and internationally, I should add. The museum's research plays a critical role in understanding and protecting our biodiversity. And at the core of this, our unique museum specimens inform this research, such as the Thylacine joey, which you'll hear a bit about tonight, preserved by Museum Victoria in its collections for over a century. The joey was used to sequence the Thylacine genome, which is an integral component of the de-extinction project, which we'll be discussing tonight. So it's my great pleasure now to introduce our host for the evening, ABC Radio National's Natasha Mitchell, who will come and introduce our guest speakers tonight and facilitate the conversation. So please make Natasha welcome. Thank you.
Fantastic. I'm very excited to be here at this event, Reversing Extinction. I want to make a special acknowledgement of the people joining us on the live stream. Thank you so much for being here, and in the room of this beautiful institution, Museum Victoria, thank you for joining us tonight. And we want to make this as interactive as possible. So use your phones or whatever device you've got here, and the idea is to use the Slido platform. So I want to weave questions throughout. We'll get kicking off with a discussion, but to ask a question, you go to slido.com. I think the information's up there, or it will be shortly, and you use your mobile phone to get there, and then you enter the code for the event, which will be up there, extinction 0610. So that's extinction 0610. and I'm going to weave your questions in throughout the night. But we are facing an extraordinary crisis. The word crisis gets overused, doesn't it? But this is genuinely the case right now. And Australia is one of the worst in terms of extinction rates. You know, we have the leading mammal extinction rate in the world. That is Australia's shame. And we have amongst the most unique species in the world. And yet we're letting them go at amongst the highest rate in the world. What a great tragedy that is. As we clear great swathes of land, as we concrete over the surface of the earth, we just don't give critters a chance to exist, to thrive in the habitats that they call home. So we humans are clearly the problem here. But could we also be the solution? That's the focus of the discussion tonight. What if we took the tools of genetic science and used them to bring extinct animals back from the dead? Is that science fiction? The podcast I host is called Science Friction. Can't help myself. Could we engineer them back into existence using the DNA from the skins and skeletons in amazing institutions like this one? How close are we to that prospect? And if we could, should we do that? Is that the right thing to do? Would the woolly mammoth and the passenger pigeon and the thylacine, would they be happy amongst us here in the 21st century? Is this their world? What sort of environment would we be bringing them back into? And would they be very lonely? Would they be very isolated? So these are some of the themes that will come up today. But very importantly, de-extinction isn't just about breathing life into the dead. It's also about using genetic science, using these tools we're going to talk about tonight to stop endangered animals from going extinct in the first place. It is about conservation, and there's some really interesting frontier science happening right here on the stage, actually, in fact. So let me introduce our fantastic speakers. We have three scientists at really interesting, exciting coalfaces. Dr. Kevin Rowe is Senior Curator of Mammals at the Museum of Victoria. Rodents are his passion, and his research has led to the discovery and description of 6 species of mammals, with several new species still to describe. Congratulations, Kevin.
Yeah, I also study shrews. We described 14 new species last year of shrews. So
What was that?
I also study shrews, if anyone knows what shrews are. But
Shrews. Andrew Pask is Professor of Epigenetics at the University of Melbourne, where he heads up the beautifully named TIGR, the Thylacine Integrated Genomic Restoration Research Lab. So his team are the team that sequenced the genome of the extinct thylacine, the Tasmanian tiger, and now they're investigating whether they could indeed bring back the Tassie tiger. They're also focused on preserving and conserving threatened marsupials. Thanks so much, Andrew, for being here. And in the very wee hours of the Californian morning, we are so thrilled to have with us Mairin Balisi, Curator of Paleontology at the Raymond M. Alf Museum of Paleontology. And what's really cool about that museum is that it's actually co-located on a high school campus. How excellent would that be? Mairin specialises in extinct North American mammals, so cats and dogs and their relatives across deep time. So we're talking megafauna like the saber-toothed cat, the dire wolf, and she's also investigating how past extinctions in the face of changes like climate change might help us understand extinction today, that deep time perspective. So get on Slido and start asking those questions. We have the code extinction 0610, slido.com, and I can see some of your questions are coming through now, so I'm going to weave them in as we go. I want to ask you all though, given that we are responsible for making animals go extinct, all sorts of species go extinct, do you think that we have a moral obligation to try and bring them back? Andrew?
100%, absolutely. I think if we have the technology now to undo some of the wrongs that we caused, I think we have to think very carefully about which species we want to bring back. They have to have somewhere to go. They have to have habitats, that they can exist in. But I think if we have the technology and the ability to undo some of those wrongs, that we absolutely owe it to those species to do that.
Kevin.
Yeah, absolutely. I mean, we know from the deep time record, from the historical museum record, the huge impacts we've had on ecosystems, how we've disrupted communities globally, and ecosystems are suffering, and whatever we can do to help remedy those wrongs and bring about healthier ecosystems is going to mean for a more bountiful and beautiful planet to live on.
Mairin, do we have a moral obligation to bring back some of the species that you study, long extinct species that have not roamed this planet for potentially millennia?
Well, I think that that's a really interesting question. So, Natasha, you mentioned that one of my focal species has been the saber-toothed cat, Smilodon. This was a very large cat, larger than basically all of the cats that we have in North America today. And I think that if we were to bring the saber-tooth back, that might cause more problems given people. So I think that it depends on the species, and I think that that's a really great point of discussion. And I also, what Kevin mentioned, Kevin, you started mentioning ecosystems as well, and I think it really depends on what about ecosystems, what about extinct ecosystems we're trying to revive.
Yeah, what would we be bringing them back into? So, Andrew, what makes then the thylacine, the Tasmanian tiger, a good potential candidate to consider in your mind to bring back from extinction?
I actually think of all the examples for de-extinction, it is the best one. So it's not just a really good one, I think it's absolutely the best one that we have. And I think that's because it's a recent extinction event, so we're able to get really good quality DNA from the specimen. So that means we're not going to bring back something that we're filling in gaps or making up, with other bits of other genomes. We're really confident that we've got an excellent complete genome build. But it's also a species that was a human-driven extinction, so this is something that we aggressively hunted and wiped off the face of the earth. And it's a species for which the ecosystem still exists, which it once thrived in. So it's an animal that if we could bring that animal back tomorrow in its entirety, you could actually put back into that ecosystem in Tasmania, and it had enormously important benefits in that ecosystem. It was this incredibly unique animal, this apex predator, that sat right at the very top of the food chain. And there are no other Australian marsupials that fulfil that same role or that same niche. And so when it was lost from that ecosystem, it caused a huge imbalance. And I think for all of those reasons, if I was to pick anything, it would be the thylacine to try and do this with, because of all of those reasons. It's really got everything going for it for a de-extinction project.
But have you seen the roadkill in Tasmania lately? So if you brought it back, is it actually going to survive those roads? It's a tragedy driving through Tasmania.
There is a lot of roadkill there, but we would hope that it would become abundant enough, like most of the other species there, that it can definitely combat dealing with a bit of traffic and people.
Right, we've got a question then from Kengo, who's 8. Where's Kengo in the audience? Hello, Kengo, or you might be online. And Kengo wants to know, how do you bring the thylacine back to life?
It's a great question. So we still can't create life where there is none. So we can't actually go to museum specimen collections, find an extinct animal and somehow magically bring that back to life. We still can't create that element of life. So what that means for de-extinction projects is you have to start with life. You have to start with something living. So the way that these projects work is you look around in nature to find the closest living relative of your animal that went extinct, and then we can set up cells growing from that living animal. And then we compare their DNA. So we compare everything about their blueprint or their set of instructions on how to build that animal. And we look for everywhere that they're different. And they're not hugely different when it's a closely related species. There's a lot that needs changing, but it's not a massive amount. And then basically using DNA editing technology, we take that living cell and we edit it and change it into a thylacine genome or into that Tasmanian tiger genome. So you're basically engineering your Tasmanian tiger
into being.
Back into existence, yeah, exactly. But brief on how it works?
Very brief, and you make it sound like you could do it tomorrow.
Oh, cinch. No,
Yeah.
this is incredibly
So
complicated.
this is hard stuff. So
Yeah.
what are the difficulties?
Really the scope of the edits that we need to make, so just going through and editing all of those changes is huge. But also we lack a lot of the basic technologies that we need to turn living cells into whole living animals in marsupials. So things like cloning, IVF, simple assisted reproductive techniques, we really can't do yet in marsupials. And that's some of the things that we have to figure out in this project. But my real passion for this project is because all of those techniques have incredible conservation benefits for marsupials right now. We know we're facing this massive extinction. We know with events like the bushfires we had a few years ago that we run the risk of losing entire species of marsupials, but certainly a lot of their diversity in just, one single adverse weather event. And if we are able to develop this technology of just taking marsupial cells and turning them back into whole living animals, then we can start to use a lot of these techniques to protect against the further loss of other marsupial species and make sure that if we do lose any of our modern day marsupials, that we can repopulate those animals once the bush is regenerated.
So many questions, so many ethical and social, environmental questions that this work triggers. Kevin, you're interested, though, in using some of the techniques that Andrew and co are using to think about how can we keep the species that are on the brink now. And so you're focused, for example, on endangered rodents, and particularly one very cute, gentle, beautiful specimen called the smoky mouse. But tell us about how you enter this conversation.
Well, like Andy said, these bushfire events and other declines in species are all reminders of what we're losing on the landscape, and we're losing in species. We're losing all this genetic diversity, and what these technologies allow us to do is when we encounter an animal through research, or even if it's had a fatality or mortality, we can try to preserve its cells as living cells so that if we need to bring back that living genetic material, we have a much greater starting point than working with thylacine that we've had in the collection for 113 years sitting dead in ethanol. And that's what our Living Biobank initiative is really about, is trying to preserve the living cells from animals in liquid nitrogen, and we can preserve them, cryopreserve them, and keep them alive for the next century to come, and so that we have access to that genetic diversity with future technologies and future needs.
So these are actually active projects happening now at the museum.
Yeah.
So a living cell biobank, there's a DNA zoo that people are driving.
The frozen zoo.
A frozen zoo.
The frozen zoo, yeah, is an initiative that started at San Diego Zoo, and it's actually pretty old. It started in 1975, and that was to set aside living cells from animals in captivity, like rhinos and other things of conservation concern. And the technology was just really challenging to do, and really could only do it in a laboratory space. And what we're trying to do, and which seems to, we're now at the point we can do, is collect cells from animals in the wild. So if we catch a smoky mouse, which we studied them, we've been studying them for more than 10 years in the Grampians, and we know that they're in these very small, narrow ranges there, and we've been tracking individuals. So when we catch an individual, and we can take a small ear biopsy, which we typically take just for DNA samples. We're now able to sequence whole genomes from those individuals, and now we can, instead of just taking dead DNA, we can keep those cells from that ear alive, those skin cells, those fibroblast cells alive, and then we can culture them in our laboratory.
No animals were killed in the making of this this cryobank.
No. So yeah, part of what we're trying to understand is the survival of these animals. We have individual males that we've seen over a 5-year period living on these sites. We had a male go away for 4 years and come back last year. So we have these marked individuals. That's part of the science, is trying to track when their survival is lowest, what are the impacts of cats and foxes on this landscape. We have cats and foxes all through the landscape. When is their predation most at risk for the smoky mice? So this is all wrapped up in the understanding the ecology, the mortality and survival of individuals in this landscape. But then as we're watching them, when we have them in hand, we can now set aside so that even when that animal dies, hopefully after a long 5-year life as a smoky mouse, we then have its living genetic material, because we know that genetic diversity has collapsed for most mammalian species in Australia.
Why is that important? Why is it vital that we preserve genetic diversity?
Genetic diversity is the stuff of evolution. It's what allows species to respond to a changing world and changing environments. But also, the way that we collapsed genetic diversity meant that a lot of that remaining genetic diversity is actually of lower fitness. So that animals suffer these genetic diseases. We see them in humans. We know what it means when you get inbred. You know what's happened to royals all over the world and the diseases they carry because they're inbred. We know that those inbreeding diseases show up in mammals. So, for instance, our first trip to the Grampians in 2012, we caught some swamp rats, one of the most abundant mammals in Australia, and those animals turned out to have this really rare genetic disease called erythropoietic porphyria. It's a breakdown in hemoglobin synthesis that leads to deposition of hemoglobin in their bones, and they get red skeletons. And we only discovered that because we collected specimens for the collection to document this work. And so we know that this really abundant mammal actually is collapsing to really small population numbers even on the landscape and losing genetic diversity and then suffering genetic diseases resulting from that loss of genetic diversity.
Just briefly, because I know you could wax lyrical for 3 days on this, why does the smoky mouse pull on your heartstrings?
Well, it's because because it's where I started when I came here. So with our first Grampians Bioscan, which was one of the many museum initiatives where we do these multi-taxa surveys of important places. We have other bioscans and bush
There's no
blitzes
romance in this answer.
Sorry,
Why,
I
why does the smoky mouse pull on your heartstrings?
because I am interested in collections and temporal changes through time. I don't have this passion for rodents really, but I have passion for collections, and I have passions for the historical record of what we as humans colonizing the world have done to populations and species. And Australia is,
And the little beings are important. They are important in ecosystems.
rodents are extremely important in ecosystems around the world, and they've been important in Australia for the last 5 million years. It's taken them, they didn't got here last, but they did get here 5 million years ago.
So I'll come back to you because I do actually want to understand how you use a living cell biobank, these living cells that have been frozen in perpetuity, how you would then go about using them to rescue a population. That's interesting. So hold that thought. But Mairin Balisi, over to you in California. We've got a question here from someone. With the world's worst mammal extinction rate, should we be focused on conserving what we have left? Does de-extinction stop us focusing on conservation? I'm interested in your thoughts on that front. And then I want to come a bit more closely to your work.
Oh, sure. Before I address that, actually, I wanted to add something to what you said about small things. So at the La Brea Tar Pits, where I was for 4 years, up until very recently.
Just describe what they are. That's a very special site. Really, in the heart of Los Angeles.
Yes. So in the heart of Los Angeles is this fossil locality, and it is arguably the best, well, I'm really biased, but I think it's the best Ice Age fossil locality. It's the best in terms of numbers of specimens as well as the numbers of species that are preserved there. And so the tar pits, they are these pools of asphalt on the landscape, and they are there because Los Angeles, even though you think of Hollywood and all of that, Los Angeles lies above a large oil field called the Salt Lake oil field. And that oil is the product of, millions of years of Los Angeles being in and out of water. So Los Angeles was underwater for a good part of its history. And now that oil, which is basically fossil fuel, that oil seeps up from under the ground. And it has done that for at least the last 55,000 years. And because of that oil seepage, the surface, on the surface, the oil forms sticky pools of asphalt. And it's not quicksand, but it's just very sticky. And so if you walk across it, you will get stuck. Humans can do that, and so did many other animals. And plants.
So it's this incredible resource for you as a paleobiologist.
It is an incredible resource. So I think of the tar pits as, this really, it's a window into Los Angeles deep time history. So it's wonderful. And the special thing about the tar pits, and what I wanted to tie into what you said earlier, Natasha, one of the special things is that the tar pits preserves these large animals, so megafauna like the saber-toothed cat and the dire wolf, which is real if you watch Game of Thrones. So the tar pits does preserve those large, megafauna, right, from the Ice Age. But it also preserves the small things. And at the tar pits, as scientists, including myself, have been starting to focus to have this ecosystem focus, so this big picture focus, more and more we're starting to narrow into the small things because, as we say, small things tell big stories.
Yeah.
Ecosystem change over time.
Well, what sorts of stories do small things tell? Because what you're observing in analysing these long extinct ancient beings is you're revealing a very complex story that even if you remove one species from an environment or you put one species back, all sorts of chaos can unfold.
Yes, indeed. All sorts of chaos can unfold depending on your species. So if you have a species that's what we call a keystone species, this is a sort of species that it might be an apex predator where, it might help control an ecosystem from top to bottom, or it might be a really key plant species, for example, that, the extinction of which would cause cascading effects like throughout the food web. So those are examples of ecosystem level changes that can happen with change in a single species. So yeah, if you think about
So give us an example. Can you give us an example of how you interpret history in this way? From fossil bones or fossilised bones or whatever you're working with. Give us a real sort of visceral sense of how you tell this story, how you construct the story of a long gone species that might help us understand what happens if we put the thylacine back into Tasmania.
Great question. So, for example, at the moment, for the past 4 years, really, I've been working not on the big animals, but on the smaller ones. So, these are what we call mesocarnivores. They're small to medium sized mammalian carnivores. And they include animals like the bobcat, the gray fox, the spotted and striped skunks, which may all sound familiar, at least to viewers in California or the United States, because those species are all still alive today. But the thing is, at the La Brea tar pits, there is a great record of those species from the time that the megafauna were still roaming the landscape. So mammoths and mastodons and saber-toothed cats and direwolves coexisted with these smaller animals. And then about, between 10 and 12,000 years ago, all of that kind of fell apart with the extinction of the large animals. So I think that this story has largely focused on the extinction, but there's also the story of the survivors, right? And it's that story of the survivors that I and my students have been studying. And so what we know so far of the survivors is that they changed in body size. So, for example, you had the gray fox and the bobcat and the badger, American badger. All of those species, decreased in body size to today. And so it just seems like everything was bigger in the Ice Age, even though these are the same species, just different points in time in that species' existence. So why is that? That work is still ongoing, and yeah, I'll be excited to tune in about that.
The bigger you are, the more vulnerable you are. Andrew, Pask, if we did bring the thylacine back, have you been giving some real attention to not just the genomics and fiddling with the genetics to make this possibility a reality, but have you been giving thought to what would happen? And this is a question from Joe. What impact would reintroducing a Tassie tiger back into the environment have on the current ecosystem? And of course, all the other species in Tasmania.
It's a great question.
And what about if we introduced it to the mainland?
That's another really good question, because they used to be abundant across all of mainland Australia. So that's a consideration you could think of, although they didn't persist here more recently, you could certainly have a think about, you know, they may be able to compete with foxes and other introduced species on the mainland as well. But in terms of Tasmania, I think, you know, what we were talking about before with apex predators and these really cornerstone species, the thylacine was a cornerstone species in that landscape. It was the only apex predator in that ecosystem. And so when you lose those animals, it does throw that entire ecosystem into imbalance. And a great example of that is something like the devil facial tumour disease. So that's the Tasmanian devil that almost went completely extinct because of this transmissible tumour. And they get this from biting each other's faces. And a longer-ristic animal is able to persist in that population, the more disease it can spread and the more quickly it will go through the population. So there's been a lot of modelling to look at what would happen if you had the Tasmanian tiger still around, eating those sick and wounded animals and removing them from the population before they spread that disease. And so a lot of those models suggest it probably wouldn't have led to the almost extinction of the Tassie devil. The Tassie devil would have been quite fine probably if it was being predated on still by the Tasmanian tiger. So that's just one example of how those ecosystems really get imbalanced with those things being gone. The other thing we understand about the ecosystems is they do change over time, but they don't change really quickly. So the fact that the Tassie devil's only been gone for 86 years, or probably about, 100 years since they were in a good number, means that, there haven't been so many dramatic changes. There haven't been heaps of other extinction events of smaller marsupials that it would prey on that would really change its ability to fit back into that ecosystem and not just survive, but we would hope that it would be able to thrive once again in that area.
And, well, I was going to add, that's one of the big contrasts with the saber-toothed cat, right? Is that the saber-toothed cat went extinct, but also going extinct were the mega herbivores of North American Pleistocene. So the only mega herbivore that was left behind was the bison. You lost horses, you lost giant sloths, you lost mastodons, and all these things disappeared, and those were what saber-toothed cats were feeding on. Correct me if I'm wrong, Mairin. And in the case of thylacine, we don't have that. We have the loss of this apex predator, but most of the prey that they would have fed on are still reside in Tasmania. And then we have this introduction of these other mesocarnivores. And as Mairin said, having apex carnivores around displaces mesocarnivores. We know that from the fossil record. We know that they have an effect on those species and control their numbers. So when you have cats and foxes on your landscape, we're spending huge amounts of money and huge amounts of efforts to try to control them, baiting, doing all these controversial things on the landscape. And having apex predators is one way to help displace and control those predators.
Initially, though, we'd only be bringing back one, then maybe two, and then maybe a family. So you're dealing with that question. You bring one back, you bring two back. A question from Joe, if we are to bring back extinct species, is there enough genetic variation for breeding in the future? That's a concern, isn't it?
So one of the great things about this project as well is the thylacine had these beautiful pelts. They're really enigmatic. Museums all over the world wanted them, but there are huge amounts of specimens in collections. There's a huge amount of specimens here at the museum, and we're accessing those. We're actually going to sequence over 50 genomes as part of this project. So we're not just going to rebuild that one Tasmanian tiger genome, we're going to sequence 50 genomes. We're going to get a really good understanding of the population diversity, and we will be engineering that back in, because the whole idea is not to bring back one, or two and have that really limited diversity, but to actually bring back a really healthy population that we would hope could re-establish themselves in Tasmania.
Yeah.
But those are all questions. That's what we're going to learn by doing these kind of things. There's lots of questions that aren't answered yet, and then by attempting it, you figure those out. You figure out what genetic diversity you need to keep species alive. That's a question we have for living species. How much genetic management do we need to keep these species alive? Where are they imperiled on the landscape? Trying to do it in the thylacine, because it gets interest and brings in new funding sources, is a big initiative that'll answer those kind of questions that we have to answer in other wildlife, other species.
So here's a question, though. We've got a question from Anonymous. The Tasmanian tiger today, Triceratops tomorrow. And I'll weave into that from James. With species such as the woolly mammoth, the Smilodon, etc., would it be more likely that they be brought back into captivity for study rather than into the wild? And that's a legitimate question, isn't it? Because many people think that this whole exercise of kind of re-engineering beings back into existence is a classic human move. It's the ultimate in human hubris, scientific hubris. Oh, well, we stuffed up, so let's just engineer them back into existence. It's sort of a techno-fix solution for what is a much more complex evolutionary history. And so that's a question that I'll put to you. Is this sort of it's great fun. It's really interesting scientific challenge. But will the animals themselves benefit? Do we know? And what about Triceratops?
Yeah, so I'll start off by saying, so there's no DNA left in dinosaur bones, so we can forget the whole Jurassic Park scenario. I'm sorry to disappoint everybody. So DNA lasts has a half-life of about 500 years. So in a couple of million years of any sample in a museum collection, there's no DNA left. So we can't go back 60 million for dinosaurs, nothing there, unfortunately, for the big Jurassic Park fans. But it means we're not going to get eaten by T-Rex, which is a good thing. But for the more recent extinctions, you can definitely apply it to anything. I think, my point on this would always be, well, we didn't stop to not wipe out these animals. We're not stopping to destroy the environment. We're not stopping to protect these animals. And so you may call it hubris, but I think this is a way of trying to reverse some of those terrible actions that we've done. And I think we would all prefer to not have to engineer animals for us to just do a better job of preserving the environments and really looking after and protecting those species, but we're not doing it. And so I think this is one way of trying to fix some of those wrongs, particularly for something like the Tasmanian tiger, hunted to extinction about 86 years ago. We didn't have the foresight back then or the deep understanding of cornerstone species, apex predators. We didn't understand how unique and important it was. But I think now we can appreciate that. Now we can see the impacts that its loss has had on the ecosystem. And now we have the technology to bring that animal back. So I think, yes, we should be doing this.
Science doesn't happen in a vacuum, though. It is a very human enterprise. And so a question from Amelia, and I'll put this to you, Kevin. How would we prevent destructive corporations from using de-extinction technology to avoid being held to account for destroying vulnerable habitats? Very interesting question, don't you think?
Well, how are we holding them to account now as they destroy habitats? I mean, we're clear felling old growth forests on a daily basis, and Borneo is collapsing into no forest left. And
but you can hear it now, can't you? You can kind of hear the spin. Well, we're sponsoring the de-extinction project to bring back the thylacine, and then it goes into their annual report, and it's all tickety-boo.
Oh, yeah. I mean, that's already happening. I mean, the carbon offsets is kind of a way that we do that, right? There, and so we're just saying, we can, ExxonMobil and other places are having to deal with the excess carbon they can. So they're trying to come up with solutions to that by saying, this carbon would have been put in the environment, now it's not. But that's the corporate shell game we're going to have to deal with in our consumer lifestyle.
But it's still enormously complicated and enormously expensive. So it's not something you could just budget for and be like, we can de-extinct 20 species. Like, no, this is an enormous cost.
Yeah, I certainly hope we see that it's like, it is really expensive, really complicated, and maybe then we'll realize, it's going to be a lot cheaper if we maintain the living genetic diversity in species. And I don't think there's anyone on this panel, there's any scientist who has the hubris to say, we don't need to worry about living things, we'll just, bring them back when we need to. I don't think there's anyone who thinks that's a viable strategy. It's much more efficient to protect ecosystems. But even though we've been clamoring about it for decades, saying that we're not really doing it effectively, and we're not stopping extinction. Hopefully this new release from the Australian government will lead towards zero extinctions, but we're still altering the landscape in massive ways that lead to decline of ecosystems.
So many fantastic questions coming in. Thank you for joining us on the live stream. And we're loving all the questions coming in, and I'm trying to keep up with them all. And I've got a question for you, Mairin Balisi, there in California. Someone has asked, and I'm interested in how your work informs this, should we be bringing back extinct habitats to go along with the extinct species? How would that be balanced with current deforestation issues? That's an interesting challenge because, of course, extinct habitats are hard to tease out, aren't they?
They are sometimes hard to tease out, but just yesterday I learned of a habitat that has been around for 1 million years, and that is in danger at the moment, and it's right here in California. It's called the Pebble Plains, and if you're from California, you might be familiar with the Big Bear region in the San Bernardino Mountains. Well, apparently there's this tiny patch of land in the San Bernardinos called Pebble Plains, and that sort of habitat has been around for the last 1 million years, and it used to be connected with Sierra Nevadas, which are our large, extensive mountain range here in California. And so yeah, to go back to that question, I think that, there does need to be focus on, and to go back to an earlier question too, Natasha, I think that we have enough resources as a civilization, I think, to focus on multiple strategies at a time. And it's just a matter of what our priorities are. So, I think that it is completely possible to work on de-extinction while we are also working on conservation, and to work on, restoring habitats, even as we preserve habitats that are currently endangered. So yeah, I guess my short answer to your question would be yes, absolutely. We need to work on restoring a habitat as well, because what is a mammoth without what is a woolly mammoth specifically without the steppe tundra that it used to live in, right? Like it would just be a fuzzy elephant.
A fuzzy. So, I mean, there are very focused efforts to try and re-engineer a woolly mammoth back into being. Are you optimistic about that, or do you have concerns and questions about that possibility?
I think it's very exciting. Yeah, of course I have concerns. I think that any scientist would have concerns. Just like I think that scientists tend to be very measured in our degrees of confidence. So yes, in that respect, of course I have concerns.
So what are your concerns?
Well, my concerns are, what is a woolly mammoth? Yeah, Andrew has talked already about the technicalities of how to de-extinct a species. And, it works better if you start from life. And if you're working with something like the woolly mammoth, like you might have to start with the closest living relative, which would be an elephant. And so is a fuzzy mammoth or fuzzy elephant the same as a woolly mammoth? So that's something that we have to think about.
I like the sound of a fuzzy elephant. I'd be up for a fuzzy elephant, but would the fuzzy elephant be up for existence?
Oh, yes. Sorry, Natasha, what was that?
Well, I just wonder how the poor old fuzzy elephant would go if it arrived in the 21st century with no other fuzzy elephants to share the planet with.
Yeah. So that's what I also think about. And, part of it's probably me like projecting my like thoughts and dreams onto this one hypothetical fuzzy elephant. But I do wonder, like, how is it to be the first of your species to be brought into being, to be engineered into being? And, I don't imagine it would be very fun. I mean, yeah, but that's very interesting. Again, this is me being human and imposing my human like thoughts and feelings and dreams and hopes onto this one being. I'm interested in what Andrew thinks about this.
I think it would be very well looked after, the first one that was brought back. I don't think it would have to.
Well, according to our
anything.
terms and our construction of what a good life for a fuzzy elephant would be.
But the Mammoth De-Extinction Project, I think, is a really interesting one. It's interesting in comparison to what we're working on with the Thylacine. Obviously, it's been extinct for a lot longer. Its closest living relative, the Asian elephant, is a lot closer than what we have for the Thylacine. So that means there's actually a lot less edits to build or engineer your mammoth that are required. But then you've got a 22-month gestation that you've got to try and figure out for your embryo. Our marsupials have about a 2-week gestation, so they have maybe 14 days-ish for most of the group of marsupials that the Thylacine was part of. And their babies are the size of a grain of rice. Not even a big grain of rice, the little arboreal grain of rice. And so that is much easier for us to figure out how to get that cell to go through to an embryo that you can then bottle feed, basically, to get back into existence. But that's very complicated for the mammoth. So I think, there are huge efforts on both fronts at the moment, and I don't know who will get there first, but it'll be interesting to see.
So a whole range of questions related to that, but I'll come to you, Kevin, first. So just back to the challenge then of using some of these techniques to keeping species back from the brink of extinction that are alive now. So how do you envisage a living biobank, the one that you are creating now and feeding with smoky mouse cells and then eventually other species as well? How do you envisage using that in a practical way?
Well, we already invest a lot in genetic management of species of concern. There's a lot of artificial assisted reproductive technologies, IVF, that are used in species in zoos. And the notion here is that we can provide more of the genetic material from the wild that can be used in this framework. So some of the things we'll store will be gametes, sperm, and eggs, working with our partners at zoos in Victoria and elsewhere, but also the potential from the biomedical technologies is to take any cell, a skin cell, and turn it into, make it pluripotent, make it like a stem cell, make it allowed to grow into any egg. And scientists, this is not science fiction, did this in 2009 with mice, with lab mice. They took the skin cells from a mouse's ear or something and were able to put those, grow those, turn them into gametes, and then put them into an early stage blastocyst. And the mouse that resulted was the genome of those skin cells. So this is the ability to preserve those cells.
So a cloned mouse.
Yeah, and this is important in like communities like in rhino poaching. So now there's just paper out this year about when a rhino is poached, they can go in and now and try to harvest the cells and protect the cells, and so that they don't lose that animal from the genetic diversities out there. And that's an issue that we've learned to do in species like rhinos, but in flagship species like that, but apply to most of the species. And museums are the only place where we actually study all the species. We have 2 million species named on Earth. Almost all of those are recorded in specimens in museums, and only in museums. And we are the place that work on all the diversity of life, and that's what we're trying to put away in our biobank. Not just a handful of flagship species, but everything that we work on, especially here in the state of Victoria.
And it's such an interesting history, isn't it? Because many of the great collecting boom of the 19th century that fed museums with collections of specimens, I mean, that collecting boom resulted in many cases in extinction. And yet now, or a number of cases anyway, and now though, those items in collections are so valuable, so important in understanding.
I don't know of any evidence that collections, building for museum collections, led to declines or extinction of species. The harvesting and over-harvesting and destruction of ecosystems led to declines, and a lot of that was documented by museums from an early stage in the early part of the 20th century, realizing that we were altering our landscapes and trying to document that change and leaving a record. I came from the University of California, Berkeley, the Museum of Vertebrate Zoology there, and the first director of that museum wrote in 1910 that the value of what we do will not be realized for more than a century, when the student of the future will want to know about the faunal conditions of California or wherever we now work. And that's where we are now. So we've looked back at those material. We've looked back at the Thylacine from 1913. We've looked back to Gould's mouse and other extinct species of rodents to realize what we lost. And now we're trying to prepare for forward when the student of the future is going to look back in a century.
Yeah, that's right. But I guess the interesting point is that these items of extinct animals are so valuable now in museum collections because they can help conserve and protect future species, keep them back from the brink. But also the museums have now have a different approach to collecting. I mean, this idea that you're building a tissue bank from living animals, a living cell bank from living animals. It's not like you're having to kill animals in order to build these precious resources to help you as museum scientists to better understand those animals.
Well, that's a complicated subject. We've published on that here in our journal, Memoirs of Museum Victoria, on the value of voucher specimens or collecting whole animals and their minimal impacts. We talked about the road toll in Tasmania on wildlife. That's like 1 million species, 1 million animals a year, and each year duck hunters kill something like 600,000 birds in the state of Victoria alone, and over the last 160 years, Museums Victoria has collected around, I'll probably get the number wrong, about 250,000 vertebrate specimens in Victoria. That's on average, less than 1 specimen per square kilometer. A lot of that density in places like urban developments of Melbourne. But the times have shifted, and there's numerous papers have been written. I gave a talk last week at the Australian Mammals Society about the value and need for collecting and the minimal impacts. But we keep wringing our hands saying, oh, we should do it. But the culture has shifted. We have to make changing decisions about what we can collect.
Could you imagine A species that you're working with, a species of rodent, a precious rodent in a precious habitat, being re-engineered. Could you imagine engineering genes into those communities of rodents so that they are protected
Well,
from
we're doing
predators?
that in a way now. We have quite a few translocation studies and programs across Australia where we're trying to move animals around to try to maintain the animal husbandry, maintain the genetic diversity. It's just another tool in that if you can say, so one of the animals we studied was the Gould's mouse, which we thought was completely extinct. It was first discovered on the voyage of the Beagle in 1837, and then last collected by our first curator of zoology, Wilhelm Blandowski, in 1857 in the northwest corner of Victoria. And then we thought it was extinct. And then we sequenced those specimens, both the ones in NHM in London and here in Museums Victoria, and they showed that species survived on a single island off of Western Australia, and it was known as the Shark Bay mouse, Pseudomys fieldi. And it turns out it lives on that species. But we also showed that from those specimens in the 1830s and 1850s, they had high genetic diversity, and that high genetic diversity at the time of their last collections completely collapsed to this single island endemic in the Shark Bay mouse. And I know the people working on the Shark Bay mouse recovery. They're putting a lot of investment trying to move them to different islands. They're trying to manage their genetic diversity, but there's almost no genetic diversity left. So that's, as I said at the beginning, genetic diversity is the stuff of evolution, how you respond to a changing climate. All they need is one dramatic change, much like the Bramble Cay melomys faced as sea levels rose and wiped it off its cay in the Torres Strait. The Shark Bay mouse, with some dramatic changes to environment, won't have the material to respond to evolution. And we now have the material in the Museum Victoria collection and the NHM London collection to say this is some of that genetic diversity that was lost. What if we can recover that using the tools we have for
And how close are
Selma?
you to doing that?
Well,
Will someone do it?
Well, I think
Is it going to happen?
with the
Andy,
de-extinction
you wanna do it?
projects, I think that's exactly what we're doing, right? We're going to engineer back in diversity.
Because
and I
you're
think-
looking at that in relation to the northern quoll, aren't you?
Right, and I think that will underpin a lot of those projects. I think that, yeah, the northern quoll is another great example of why it's so important to develop these technologies. So in the process of engineering our thylacine, we have to make the technology for DNA editing a marsupial and then bringing that animal back. And one of the key projects we're working on from the get-go is with the northern quoll. So the northern quoll's, this beautiful little cat-sized marsupial. They're going extinct in northern Australia because they eat cane toads, the introduced toad, and they're toxic, and that just kills the animal and they die. But we know that animals that naturally evolved with cane toads or with that group of toads, they have all evolved resistance to the toxin, so they can eat those toads and they're fine. And it's just one single change in their genome that you need to make an animal resistant to cane toads. And so we can engineer quolls to be cane toad toxin resistant with a very, very simple edit. And then not only will that save the northern quoll from extinction, but they can actually then eat cane toads, which are this terrible ecological disaster. They can actually help control the spread of cane toads, which would be a phenomenal thing. So you can actually, use this technology not just to preserve our animals, but actually to act as biological controls, to actually help, deploy, better ways of looking after these species. And this is a change that we know naturally evolves in an animal's genome if they have long enough. The unfortunate thing with the northern quoll is it will go extinct before we have another millennia for it to evolve that toxin resistance. So it's just a way, really,
Could you
of
re-engineer it?
speeding
so that
those things up.
Interesting.
So-
So on that, because there's hundreds of questions and I need to get to them. But relevant to that, would this ethically affect humanity? Would this give us a God complex? Is that a good thing for society, asks Sai. And then also Peter asks, what are the ethical issues around creating what is in reality a new life form? Part Dunnart, part thylacine.
So I think, with the first bit, again, I'll say, well, we wipe them out without stopping to consider were we acting as God, and, we change the landscape around us, we're changing global temperatures, we do things that play God all the time. So I don't see that this is actually good God. This is like New Testament God, not Old Testament God, when we're trying to bring some of these things back, right? So I think, we already have a terrible God complex, and at least we'd be applying it in a good way.
Mairin, is this good God or bad God?
I was thinking about it more, is it lawful good or is it chaotic neutral or, those are the because I do work at a secondary school, I'm exposed a lot to memes and whatnot. So
Well, I'm interested in your perspective, though. This idea that we can, I mean, you're studying species that are long extinct, but they evolved over millennia through a fascinating process that took time. So this idea that we can just, well, re-engineer beings back into existence, what do you make of that? That's at a very different timescale with all sorts of potential unintended consequences that we can't necessarily predict.
yeah, I think that all of these are great questions, and I think that you make a very good point of the accelerated timescale. So, yeah, I mean, the world is changing at an accelerated timescale at the moment, and so I do think that it makes sense to think about, long-term solutions, but also solutions that might help in the short term or in the shorter term. Um, so yeah, I think I'll go back, or I will go back to my answer from earlier that, why not both? Why not try to tackle the issues at multiple different scales? So, like those of us, like there are individual scales, right? Like there are, riding a bike to work every day, which I did for the last 10 years. And then there's the, like corporate level solutions. Like I think that we can I don't see why we shouldn't try to attack it at multiple different scales because there are um, multiple different problems all happening at, faster than these are multiple different selective pressures that are happening faster than they have um, over the last millions of years, the last millions of years that I have studied personally.
Kevin, would reintroducing the thylacine be a death sentence? This is a question from someone for already threatened small mammals. This is one of the potential unintended consequences.
Well, probably not, but we don't really know. And there's a lot of this leaping to we're just going to throw a big pack of thylacines out there.
Yeah.
This is going to be a lot of research. It's going
Yeah.
to take time to figure that out. And I think the examples that we've done reintroductions of extinct species, locally extinct species like timber wolves in Yellowstone, are a great example of how they were the lifeblood for the whole ecosystem from the highest peaks to the river systems there. And I encourage you to look into that kind of work, the trophic cascades that you get when you reintroduce an apex predator. Because I'll tell you right now, the threatened small mammals, the smoky mouse included, are just imperiled everywhere by cats, foxes, and other introduced herbivores and predators that are just destroying their habitats and consuming them. And it's something we don't know how much to do about it. Right now, they're in Western Australia, they're trying to build like an automatic cat killing box called the Thylacine that shoots a bait. It recognizes the cat, shoots bait at it, and kills it. So there's lots of different technologies that we say. As Mairin said, we're a civilization big enough to come up with multiple solutions. But dealing with feral predators and feral cats in Australia is much bigger concern for me than bringing back a native predator like a thylacine. It will not.
So on that question, though, another question from someone, could thylacines help tackle the problem of feral dogs and cats on mainland Australia as well?
That's one of the big ideas, right? And there's a lot of really important research on how dingoes and naturalized dogs affect other predators like foxes and cats. And there's lots of really sophisticated research there in ecological systems that's showing that having dingoes around can help displace and control foxes. And almost certainly a thylacine there would help displace it. They'd also help remove the Tassie devils with facial tumor disease and keep populations healthy. This misnomer of predators are eating prey and hurting prey was something we learned about in the early part of the 20th century in wildlife biology. Aldo Leopold and other people who were out there, their job, like here, was to shoot predators. So we were shooting thylacines because we thought, it'll make for better ecosystems, it'll protect our sheep. And they realized in the early 20th century that shooting a puma did not mean for better deer. It meant for worse deer and worse deer populations, and they declined. So reinstating our predator populations is really critical to saving our small mammals and prey species.
So many questions here. I just want to ask every single one of them. Is it really necessary, someone asks, to reverse extinction? Maybe the nature has made its peace with the dodo bird being extinct. Why does this deserve priority public funding?
So I don't. None of the work I've had has had public funding for trying to bring back the thylacine. So this was a philanthropic donation that was given to me, and also biotech companies that have wanted to invest in driving this technology. So I think no public funding for this, and it's brought millions of dollars to conservation biology for marsupials that would never come through the public funding means. So it's actually been a really effective way of attracting more money to helping the plight of other species, which I think is a really important consideration of this work.
So are
But
people drawn to the novelty of funding this?
Yeah, I think
But then
they
it has
really-
this sort of flow-on
That's it.
effect.
They really want to see these species brought back, these cornerstone species. And I think we have to develop all these technologies like the DNA editing, like the cloning, for marsupials along the way that will have enormous conservation benefits for what we have left and protecting those animals that we have left, as well as one day, hopefully, trying to bring back some of these species that we've lost.
Here's a question as we approach wrap. What safeguards would we need to put in place to prevent the exploitation of any species we bring back from extinction? That's a really interesting question. And just taking that to a kind of further degree, here's a thought experiment. As technology advances, ask someone, how do we ensure that rogue nations and corporations won't abuse the technology, that is, bring back Neanderthals to exploit as a form of labour?
People have talked about de-extincting Neanderthals, so that is definitely something that people have proposed in
They
the
certainly
past.
have.
And we should
Yeah.
note Svante Pabo just won the Nobel Prize
Right.
in medicine,
Yeah.
principally the sequencer of the Neanderthal genome. So this is cutting edge science.
Yeah, but I mean, we have the technology. It's around today to do DNA editing, to make genomes, completely artificial genomes from the ground up. You don't even have to start with, something that we have. You can actually create whatever you want. So that technology's there. It's available to anybody who has the money and the wherewithal to use those tools. So I do think it then falls on society to think about what do we want to see done with this technology? Where do we think we need to draw the limits with what we want to do? I wouldn't recommend bringing back dinosaurs if we could get DNA from them. We can't, but if you could, obviously this is not something that there's a habitat for, that there's a good reason for bringing back other than we would want to see it.
Well, Rod
So,
here wants to
yeah.
bring back Grandma.
That's a lot easier. I get asked about cloning humans all the time. I'm like, we've been able to do that for years.
Yeah,
That
as
is
Rod touches
definitely-
on, when
that's
we talk
easy.
about playing God,
Yeah.
we already are playing God in this way with human medicine.
Yeah.
And we're spending huge amounts of money globally on trying to grow patient-specific organs from their own cells, and that's going on as part of a way to have us live longer, allow us to survive our own individual extinctions.
Yeah.
And what wildlife biologists are suggesting, and what Andy, and I don't want to speak for Andy, but what we're suggesting is that bringing those tools to bear on wildlife and all the other species in biodiversity, the other 2 million species, and we estimate 10 million species on this planet that we share.
An interesting question for you, Mairin. Do you think that the public will ever actually accept a species brought back via scientific means? And then someone else has asked, what impact will de-extinction have on evolution? And I guess I'll couple those by asking, there's this idea of the natural, which is a little bit of a furphy, really, because what is natural in this on this planet of ours, given the breadth of our impact? But what are your thoughts about that?
Yeah, so I think that, natural and artificial, those terms come with value judgments, right? And so does, native and introduced, like things like that. Yeah, the question is, should those value judgments be there, or should species be evaluated based on their function in the ecosystem that they're in, or the ecosystem that they're engineered into? And so, sorry, Natasha, I actually, may I ask you to repeat the first part of that question? I think
Would
that
the
was
public accept
would the public
re-engineered
accept-
species, I guess?
Well, I think it depends on what the species is.
Yeah.
But, we have engineered livestock and plant crops and things around us all the time. So I always think, we already have, as a globe, accepted these things. They're out there. And now we'd be applying those technologies to native animals, at least, for benefit, rather than just for commercial gain or for, feeding the starving world or those sorts of things.
Yeah, so going back to thylacine, I have heard that there are a lot of people who would like the thylacine back, and who, there have been hundreds of sightings over the last century. And so I
None
imagine
credible.
Yeah.
yeah, yes. Disclaimer, non-credible. But I imagine that, given that interest in that species, I think that people, people might be more likely to accept the thylacine. Um, but generally, I think that, there is a value judgment, I think, on herbivores versus carnivores as well. So, if it's an apex predator, it would be not super accepted. I mean, think of so here in Los Angeles and Southern California more generally, we have mountain lions. They're still alive today, and these mountain lions are they have been here for tens of thousands of years. There are fossils of them preserved at La Brea tar pits, and they are still, they still roam the landscape where humans live, from the city of Los Angeles to, out in Death Valley.
Yep.
And so just thinking about the public's reaction to a mountain lion. I mean, in Los Angeles, we tend to like our mountain lions as a charismatic, species of conservation concern. But we probably still would not want to be outside hiking and run into a mountain lion, right? And so, yeah, there's this. So apex predators, even though they have great potential to be a cornerstone species or a keystone species, I think that engineering them, other than the thylacine perhaps, but generally engineering apex predators probably would end in disaster in more ways than one.
It's a very interesting question. We need to wrap now, but I'm just going to throw one more question that someone's asked about, is extinction a reversal, a way to adapt to climate change by bringing back species better adapted to our changing environment? Kevin?
Yeah, I mean, I think recovering species and recovering the genetic diversity that's lost in species to allow them to respond to a changing world is part of what's behind reversing extinction and de-extinction. So yeah, I definitely think it's part of dealing with a changing world. The less diversity we have, the less we're able to respond to a changing world.
Andrew?
Yeah, there are projects at the University of Melbourne now looking at engineering coral to survive warming temperatures in the ocean and also acidification in order to make sure that the reefs survive, as a hub for so much biodiversity. And so absolutely, this is one way forward that we can engineer to maintain some of our biodiversity. Obviously, it's not ideal. We don't want to have to engineer our animals in order to survive. But as if this might be the thing that we're faced with. We either engineer animals and allow them to be able to survive and keep that biodiversity, or they'll be lost forever.
Do you have a sense that this might happen, that you might bring back the thylacine within your lifetime?
Yeah, absolutely. I really do.
That's a yes, absolutely.
Yeah.
So what if we just narrow the time frame a little bit?
Yeah, people always want to. I reckon in 10 years' time we'll have edited cells. And I would hope that we are at the point then that we can actually turn those cells back into whole living animals. So I'm hoping a decade to be able to start to bring back the thylacine and see, and then you would have to study it though for 20 or more years before you would release it in Tasmania.
So
right? So it's not like you bring it back and let it go. It's still a long process.
And these two questions, I'll combine them as the last question. Sally asks, what's the importance of learned behaviour when reintroducing? Would prey know how to avoid predators? Would predators hunt well without learning from adults?
Yep.
And then also Peter Brown asks, what are the animal husbandry challenges of raising a thylacine joey to a fully functional adult? Who are the animal husbandry partners for this project? So you've got to get that sorted out within 10 years.
Yeah, so the animals really have a lot of behaviours pre-programmed in their brain, and a great example of this is domesticated cats, right? They've been domesticated for thousands of generations, but if they see a mouse run in front of them, they're going to run and pounce on that mouse. We can take orphaned animals out of the wild and hand rear them, and they will still go out and hunt and have that same role in the ecosystem as the original animal. And so we're really confident that, if you bring back a thylacine, it's going to know what to do, it's going to know what to eat and how to hunt back in that environment. Marsupials don't have enormous brains, they have pretty small brains. And so that probably means they have a lot more pre-programmed and less learnt behaviours that we think actually bode really well for the thylacine being able to figure these things out. But until we do it, we don't really know. And that's one of the things that we will study intensively.
So
And I
interesting.
actually have something really relevant to contribute here. So related to cats, the mountain lions that I mentioned earlier. So again, they have been around Southern California for tens of thousands of years. They used to coexist with the Western horse. So horses, as you know, the whole family of horses, equids, are from North America originally, millions of years ago, and then they went extinct at the end of the last ice age about 10,000 years ago. Mountain lions persisted, so that mountain lions lived without horses on the landscape for, for millennia. But then now we have horses again imported from Europe, and we have donkeys as well through domestication. And there are donkeys now roaming the landscape with these mountain lions.
Aha.
And the donkeys are definitely
Dinner.
introduced. Somehow, mountain lions, we have just documented mountain lions preying on these wild donkeys. So there is, yeah, I think that there is some behavior that seems to be, mountain lions were able to prey on horse-like animals even after millennia of not having seen one. So,
Ice Age behaviour reincarnated. What a fascinating discussion. And the questions have been absolutely fantastic. Thank you so much for asking them. And thank you for joining us on the live stream. If you posted questions, we're so glad to have had them. And hopefully I got to kind of a representative sample. I tried to kind of bring together various themes that you were asking in some of the questions that I was asking too. A real treat. And I think what we've learnt tonight is that museums like Museums Victoria are such a powerful resource for conservation in all sorts of interesting ways, both in terms of their old collections, the hundreds, thousands, millions of specimens that are held in facilities like this one, but also in the kind of frontier science that they're working on with collaborators like Andrew at universities and more widely. So that's a really exciting sort of relationship, I think. And also Zoos Victoria. I mean, zoos are now right at the frontier of this work, collaborating with the museum as well. So it's all the institutions are changing and evolving as the challenges evolve. I want to encourage you to fill out the event survey. You go into the running for a $50 museum store voucher, which is a fantastic thing. If you bought ticks and you have viewed the live stream, I think you've got access to this for another 2 weeks. So that's fantastic. And there's another Future Forums event, First Nations for Water Justice, on Tuesday the 8th of November here at the Melbourne Museum. Check that out and spread the word for us. It's been such a treat to talk to the three of you. I think we've still got Mairin Balisi, thank you so much for staying up until
2:30.
2:30 in the morning.
oh no, it's only 1:30.
Oh,
Okay. It's been
Thank
so
you so much for having me.
You're getting a very special applause. And thank you to Andrew Pask and Kevin Rowe. And thank you to all of you for being part of the Future Forums here at the museum. Cheers.